So in the second podcast, we're going to be talking about telomeres and DNA repair. So we've just talked about the steps of the cell cycle and how during S phase DNA synthesis occurs. And in this video, we're going to talk about the ways to replicate the end of linear chromosomes and then to correct DNA errors and repair DNA damage. So there are some challenges with replicating DNA in eukaryotes, and we've talked about one of these challenges, which is that there's just more DNA. So just to compare the genome sizes, and I mentioned this in the last video, E. coli, which is a kind of bacteria, have 4.2 million base pairs. Moving up from there, yeast, which is a single-cell eukaryote, has 16 million base pairs. So this is 4 times as many base pairs. And so it would take quite a long time to duplicate its DNA. And then on a much greater scale, humans have 3 billion base pairs. So this would just take a really long time if we were starting in one origin of replication. So the solution here was multiple origins of replication for these eukaryotes. The other problem is linear chromosomes, okay? And I'm going to talk about why this poses a problem. So let's take a look here at our 2 parental strands, okay? So we have our leading parental strand that goes from 3' to 5', and so the DNA polymerase will make then 5' to 3', so it'll move in the direction of the parental strand. And then here's our parental lagging strand. That's 5' to 3'. So the telomere or telomeric regions are regions at the end of eukaryotic chromosomes. So this is what happens at the end after we've finished duplicating the rest of the chromosome. And in the parental strand, DNA polymerase will move 5' to 3' and finish copying the leading strand. And it'll have a daughter strand. And in the lagging strand, we have our RNA primer added by the primase. And then we have our Okazaki fragment, so that the DNA polymerase fills in. Now, the next step is that DNA polymerase 1 and DNA ligase will work together to remove the Okazaki fragment. So the final Okazaki fragment is made and the primer is removed. Now, here's the problem right here. This is unfinished, okay? And the DNA polymerase can't add anymore because there's nowhere to stick a primer. So this in theory would pose a big problem because single-stranded DNA on the parental strand right here, on this lagging parental strand, would be eventually degraded. And this would shorten the chromosome by 50 to 100 nucleotides at each cell division, and eventually chromosomes would disappear. So this is a huge problem for replicating linear chromosomes. So how does the cell solve this problem? Well, let's talk a little bit more about the kind of DNA that's in the telomeric region. So first, there are no genes in this region of DNA, so nothing that encodes for a protein. Instead, we have this sequence, TTA-GGG, and it's repeated thousands of times in the telomeric region of DNA, okay? And what this DNA does is it allows for an enzyme called telomerase to replicate the telomeric region of DNA. So let's talk about how telomerase does this. So I'm going to start right here and explain kind of in the middle of the figure and explain what we're looking at. Okay, so this is telomerase. So this is an enzyme. And so here's the protein part of the enzyme. And telomerase, also within the protein, has its own RNA template, okay? So its RNA template is CAAUCCC, okay? So now remember, this is the exact complement of the telomeric region of DNA, okay? So remember, let's go back a slide. We said TAAGGG, okay? So here's TAAGGG, okay? So this RNA guides the telomerase to the telomeric regions of DNA, okay? So here's our unreplicated overhang, and the RNA primer in telomerase guides it to the telomeric regions of DNA. And what happens is then, so it binds here at the parental strand, and using its own internal RNA template, it extends the strand, okay? So we're actually extending the parental strand first, okay? And it's going to add a few, and then it's going to move down a few more times, and it's going to repeat this multiple times, okay? And it's basically going to then grow the parental lagging strand. Then, once it's repeated a few times, the primase, the original primase, is going to add a RNA primer, and then DNA polymerase and the clamp are going to come in and fill in the rest of this sequence, okay? So this is exactly how we are making sure we don't delete the ends of our chromosomes at these telomeric regions. So telomerase in our cells is primarily found in gametes, and we'll talk about the difference between gametes and somatic cells. So these are the ones that are sperm and eggs, and they're found in stem cells. So in our cells, right, somatic cells, and most of the cells in our body, these are somatic cells, they actually normally lack telomerase. And so the chromosomes do progressively shorten as individuals age, and this is thought to contribute to the process of aging. And there's a lot of really interesting work that's going on right now looking at telomerase, and so far evidence has shown that the number of cell divisions is limited by the initial length of a cell's telomeres, and that adding telomerase to cells in culture actually helps them to continue to grow and divide longer. So could this be used to combat aging? Alternatively, cancer cells have been shown to have active telomerase in their somatic cells, and this may help them grow and divide. So perhaps targeting telomerase in cancer cells may be a beneficial therapeutic. So that's really interesting ongoing work. Okay, so next we're going to shift gears and talk about another problem that exists with DNA synthesis, and that's mistakes. Okay, so DNA polymerase adds nucleotides at the speed of a jet engine, and it actually has a really high base calling accuracy. So it only makes a mistake 1 in every 10 or 100,000 bases, which is pretty impressive. So it's if you've answered 100,000 multiple choice and you've made one mistake. So it's pretty accurate. So here's this green box is going to represent our polymerase, our RNA or DNA polymerase, okay? And it has an active site in the enzyme, which brings in a base, right? And if this base is incorrect, okay, so right here we have an A on the parental strand that's pairing with a cytosine, okay? And so if this were to happen, this would basically trigger the DNA polymerase's proofreading activity, okay? So the correct base pair has a distinct shape, and these pairs are energetically favorable. So it's going to be hard for DNA polymerase to move on from this site, okay? So what's going to happen is this base with this phosphodiester bond here is going to move into a site in the enzyme that's called the exonuclease site. And once that base pair is in that site of the enzyme, the phosphodiester bond is going to get cleaved, okay? So this is an exonuclease because it's clipping out a nuclease. And it's removed. And then thymine, which is the correct base pair, can bind to the active site where adenine is, and this will the polymerase will form a phosphodiester bond here, and the polymerase will keep moving down the strand in the 5' to 3' direction. So, as I mentioned, the DNA polymerase has its own proofreading activity, so that helps make sure that mistakes aren't made. But sometimes the DNA polymerase, it's moving really fast and mismatched pairs are left behind. So there are other enzymatic property or enzymes in cells that can correct these mismatch sites. And what happens is these are repair enzymes. They recognize a mismatched pair, right? Because a matched pair would have better geometry. They remove that section of the newly synthesized strand that contains the incorrect base, and they fill in correct bases. And if you're interested in this concept, perhaps you can learn more about it by taking a course in genetics. What we're going to talk about next is DNA damage that occurs outside of the replication process. So DNA can be damaged throughout our lives due to sunlight, UV light, chemicals, X-rays, and we're going to talk about DNA damage that typically occurs with UV light. And so UV light and some kinds of chemicals can actually cross-link thymine bases, okay? So here is one strand of DNA with two thymines attached to each other by a phosphodiester bond. Now, normally these thymines would bind to adenine via hydrogen bonds in an anti-parallel strand. But if exposed to UV light, these bases will cross-link, and this forms a thymine dimer that produces a kink in this strand of DNA and blocks DNA replication from occurring. So the cell has enzymes that can fix this, and the name of this system is called nucleotide excision repair. And this is a protein complex that can recognize these kinks. So here's we've exposed this DNA or these cells to UV damage, and this kink is detected. And these enzymes come in and make cuts in this region where that we have these thymine dimers. So it'll cut it out or cut it and excise it. So this is the nucleotide excision. So that's one part of it. And this also involves the helicase to unwind this region so that these fragments can be removed. And then DNA polymerase will come back in and fill in the gap from 5' to 3' with undamaged DNA. And then DNA ligase will come in and seal up these regions of this newly synthesized DNA strand. Okay, so this is another way DNA repair can occur over our lifetimes. And when we talk about the cell cycle, we'll talk about checkpoints that exist in the cell to make sure that cells that have damaged DNA can't divide. So that's it for this video. To summarize, telomerases lengthen linear chromosomes so that DNA on the lagging strand doesn't disappear over time. The DNA polymerase is accurate and has proofreading activity, so it has an active site to add DNA to a parental strand. It also has an exonuclease site. And if you mutate the exonuclease site, you'll get more mistakes from the DNA polymerase. And then the cell actively surveys and fixes damaged DNA. So that's it for this video, and I look forward to seeing everyone in class. Thanks, bye.